Everyone, myself Zakaria Ahmed, Technical Assistant Biotech Hub Bahana College. Welcome all here to our third day of that 3-day online hands-on training program on bioinformatics, which is organized by Institutional Biotech Hub Bahana College, Jorhat. Thank you. Now I request Ms. Malika Barua, Laboratory Assistant, to introduce our resource person, Rafiul Amin Laskar sir. Thank you. To Ms. Malika Barua. Thank you so much, Ms. Malika Barua. Now I request our today's resource person, Dr. Rafiul Amin Laskar sir, to start the session. Thank you, sir. Now I like to hand over the session to you. This session is all yours, sir.
Using my mobile, I think that will be better. So now I am audible.
Yes, sir.
So thank you for this opportunity to present in this DBT bioinformatics training program, and also I would like to apologize for this delay because there is a program going on here in my college, and there are some few guests are there. So because of that, I was involved in that program. So quickly I will try to start. My voice is clear, na? Or is it echoing?
So it's echoing. মোবাইলৰ পৰা চাউণ্ডটো লৈছে কাৰণে হেৰি হৈছে।
Now it's clear or no?
Yes, sir. Now it's clear.
Now it's clear, na?
Yes, sir.
So I'll try to use both this laptop and my mobile to do this session. Because of some, I think in my laptop this microphone is not working, showing that it's on, but voice is not audible. So quickly to start with today's this presentation, I'm sharing my this slide first. So also I would like to thank everyone, all the participants, all the colleagues who joined today to discuss on this topic. That is, I think we all are aware of many things about this, the basics of this this primer and PCR and all those things, because in our syllabus or theory classes we have studied many of these theories, like why we do PCRs, why we need this amplification of different genes and all those things. And then also about the primers. The primers are also not new for us, because in whether it is replication or in any other biological this things, we already used this term primer. So today
মাইক থকা ফোনটোত কল আহি গ'ল। ছাৰৰ মাইক থকা ফোনটোতে কল আহি গ'ল, সেইটো কাৰণে কাট খাই গ'ল চাগে, মিটিংখন কলত আছে। জইন কৰিব চাগে ফোনটো জইন কৰা আছে। Sir, your voice is not audible. অঁ নহয় মোবাইল দি কানেক্ট কৰিছিলোঁ এতিয়া
Now audible, no?
য়েছ ছাৰ
Sorry for the disturbance. So, in this topic, I have used one example. Basically, we'll do the hands-on part. That is our main motive or main objective for today's program. Uh, few basics about the primers. So we'll start with this. Suppose a few questions means why we use this primer designing, or what is the importance of this primer designing. Any participants, if you have some queries or some questions in between my presentation, you can discuss that in the chat box. I will keep the chat box open to see what is going on. So first, I would say that what is the importance of today's presentation, or what is the importance of topic of today's presentation, is that since designing, when we are using this designing, that means whatever we are creating or whatever we are producing here, the primer, it is in our control. Means we are designing it as our as per our requirement or as per our need. Na? So how can we be, some questions like how can we be only to the gene? So since we are amplifying the specific target genes, na? So since we are using this PCR mechanism to amplify some specific target genes for our genetic research or any genomics research, so our target genes, we have to use or we have to design a primer which can bind exactly to the gene of interest, whatever the gene of interest. So that is one of the objective of designing a primer, because we have a large genome. So if you consider any genome or any species, any plant or animal genome, so the genome is a very big, large entity, or it contains many genes or many nucleotide sequences, thousands of nucleotide sequences, I would say. That we have to design a gene which can exactly join or exactly bind with the specific target gene which is present in a large genome. So that is one thing. And the second thing is that primer's performance. No? So if you could test a primer's performance before entering into the lab, that is another importance of primer designing, that before you are starting a wet lab experiment, or if you are starting an experiment with the primer, if you can test that your primer or whatever the primer you have designed, it can really work or it can really bind with a specific target gene. So in that way, it is useful to design a primer. It is useful or it is helpful to design a power. And then how do scientists quickly check whether a primer is specific? So we need a primer which is specific, stable and free from dimers or any other the things which is problematic for PCR amplification, like TM or like any other errors in our nucleotide sequence, which can really really impact the overall PCR performance. PCR performance means overall amplification of the target genes. So these are some things we have to keep in our mind. So PCR, we all know. So it is a polymerase chain reaction. Is a molecular technique used to create millions of copies of a specific DNA segment. So this is a simple or here we use a few techniques, natural annealing, extension. So all these things in a thermostable DNA polymerase, so which is thermostable. So I am not going into detail with the PCR, how PCR works and all those things. What I am concerned here in today's lecture is that for PCR, our primary requirement is to have a proper primer to which can work in a PCR system, or which can be useful to run a PCR by a specific target genes. So 1 minute. So as I was discussing, our topic, our discussion for today is today. Our discussion for today is about this primer designing. So PCR is for the basic requirement for PCR is having a very good primer which can bind exactly into the specific target genes. So from there on, I am using this NCBI primer blast to design a primer which can exactly bind with this, exactly can be used for the PCR amplification. So what is bioinformatics? So it is a basic things you all know that it is an intersection of biology, computer science and data analysis. So we all know that in bioinformatics we are basically using all this or you can say we have a interdisciplinary approach of biology, computer science and data analysis. Together we are getting some information about any biological things or biological object. So importance in modern molecular biology and genomics. So in genetics or genomics, we all know that bioinformatics have lots of use in today's world. So then you have application in drug discovery, evolutionary and agriculture, diagnosis. So all this usefulness are there. So objective for today's session, mainly in the hands-on. So kindly be there with me for this hands-on training session where we will perform some primer designing. So that is our main objective. So understanding basic of bioinformatics tools, learn principle of primer designing, and then finally perform hands-on primer designing using. So during our hands-on primer designing, you will all get the idea about how we are doing everything. Means whatever we are discussing in the theory part, it will be clear in during the designing part or in the hands-on part. And finally, what is very important is validate and analyze design primers in silico. So finally, whatever the primers you have designed, we can design thousands of primers. That is one thing. But finally, to validate and analyze that primer, whether this primer is useful or not, that is another important aspect of primer designing. So primer designing is mandatory for. It is not in the full screen, na? So primer designing is mandatory for PCR unless a ready-made validated primer already exists for our target. So what we are saying that if there is no existing primers which you can buy from the anywhere or the sequence of foods is already known, so primer designing is mandatory. So PCR can only be run or any specific target gene can only be multiplied or only be amplified only when you have designed a proper stable primer. So that is the importance of primer designing. So if we don't have proper primer, so we cannot do any of this research activity on genes or genomics and all those things, genetics. So PCR needs two specific primers to define whether application. So I am not repeating the points. You can just get the idea. But the basic idea is that without a proper primers, so without a proper primers, the reaction cannot target the correct DNA regions. So our main objective of PCR is to target a specific gene out of the whole genes. Otherwise, you will not get a band, proper band or single stable band in after doing this PCR amplification. So to amplify a specific target gene, so we need to develop or design a proper primers, proper primer. And poor and random primers lead to no amplification and wrong products. Therefore, primer cannot work. PCR cannot work correctly without good primers. Designing them is required when stable. So we have one NCBI primer blast which is available freely, which we all can use to develop or design primers. So we all know that in NCBI database, we all know about NCBI database and all those things. So in the same NCBI database where we do blast and other types of genes and other things, proteins and RNA, all those things, mRNA. So there we have also a primer designing, primer blast and primer 3, which is integrated in the NCBI database. So the primary function, if you say design primer, so what is the use of this NCBI primer? Since we basically today we will do hands-on activity with this NCBI primer blast. So the primary function of this primer blast, NCBI primer blast, is to design PCR primers automatically based on input DNA sequence. So whatever the DNA sequence you will provide, we will see in the hands-on activity how we will provide the DNA sequence. But whatever your input DNA sequence, based on that input DNA sequence, it will provide you different types of primers or different useful primers which you can use to amplify this input DNA sequence. So input DNA sequence here means it is your target DNA or target gene sequence which you need to be amplified for your research and other activities. So I am not going to the use of or activities or research area of doing amplification of genes, and after amplifying the genes, what are our objectives? That is another part. But here we are discussing about for amplification we need PCR. For amplification of any gene we need PCR, and for PCR we need a primer. So proper amplification of a target gene using your PCR depends on a stable or very primer, we can say, very primer which can bind in the specific target gene out of the whole genomes you will provide as a source material. Then you have avoid primer dimer. So there are some problems or issues during with the primers. So sometimes you will find that you have designed a primer which contains dimers or hairpins. So these will produce or create problems during PCR amplification. So your the primer must not contain any type of this dimer or hairpin issues. So allow customization. So using this NCBI primer blast, you can avoid such problems with the primer. So if you are using NCBI primer blast, you will design a primer which is devoid of any kind of this type of dimer or hairpin issues. Just 1 minute, 1 second. These are some of the identified problems or when our primers may have, and if we are using this NCBI primer blast, so we can prevent this type of primer design problems. So there sometimes if you start preparing primers, or if you already did this type of research activities or created some primers for your research activities, so you have already found that there are some problems which we usually get like primer dimers, hairpins, low specificity, incorrect melting point, so extreme GC content. So I have listed few here, and there may be many other problems in our which we have to avoid during our primer production. So these are nothing but some problems which we need to avoid during our primer designing. So in our primer design we have to avoid these problems. So why we need to avoid these problems? Because each and every different categories of these problems can create some problem during PCR formation, PCR amplification. So in our PCR amplification, it can create some problems. So I will not go into detail discussing all these problems because with the help of this NCBI primer blast.
Attendance condition.
एक minute.
Alright. data.
We all know that this is a era of DNA sequence, because why we are calling it this era of DNA sequence, because with more and more our understanding, the more and more we understand our genetic data or genetic sequences or whatever the genome we have, different types of genomes from different species, our knowledge about real biology or real biological information which we do in our bioinformatics. So we can, with the help of this bioinformatics, we have now lots of data, lots of information about our DNA sequences. Earlier we were able to code, or we have some coding sequences of our DNA. Now based on those codes, we have developed, or we have used bioinformatics to gain lots of insight into this genetic data which we already have. Then sequence, so you have role of biological database, we have different types of database nowadays. So most commonly we use NCBI database, then sequence retrieval and analysis in research. So there is another this thing. Then primer basics. So what are the basics of primers? I am now coming to the topic, because if we go into detail about PCR and genomics, that will take more time. So for primers, so what are primers? So it is role in PCR and sequence cloning. So if you see that what is the primers, it is used in the so role in PCR and sequencing and cloning. So ideal features, what are ideal features? So ideal features will be length 18 to 25 base pair. So the primers which we will produce, it will be 18 to 25 base pair. So ideally it should be 18 to 25 base pair. Sometimes it can be more than or less than that, but the ideal will be 18 to 25 base pair. Then GC content. GC, we know that guanine and cytosine content, which is very important for any kind of DNA sequence or stability of any kind of DNA sequence. So it will be 40 to 60%. There must be 40 to 60% GC content. So temperature that should be 55 to 65 degrees. Avoid secondary structure and repeat dimers. So we have to avoid secondary structures and repeat dimers. So primer design workflow. So how you will design the primers? So we have to retrieve DNA sequence. So initially the first step we need to do is to retrieve DNA sequence. So from where we will find this DNA sequence? From NCBI database. So from NCBI database, first step is to retrieve or to find out the FASTA format DNA sequence database. So any gene, so what will be your target gene? Any gene which you are targeting for your research, that can be your DNA sequence, and from that DNA sequence or from this gene sequence, you have to find the FASTA format which is available in the NCBI database. You will find in the hands-on technique, you will find again this how you can get this FASTA format, and then select target region. So out of this whole, you have to select the target region and set primer parameters. So that is another important thing. So you can set what are the parameters or what are the requirement of your primer. So in the database or in the designing, you can give some parameters that such and such things we need in our primers, and then use primer blast. So that I will do in the hands-on, that we will finally use this NCBI primer blast and then validate specificity and export this primer pair finally. So primer blast, see this primer 3, this design, and blast means specificity check. So if you see this NCBI primer blast, it is a combination of two functions basically. One is primer tree, which is used to design for design primer, and another is blast. Blast we know will know all do for specificity check. If you give a specific DNA sequence or specific gene into the this NCBI database for blasting, and it will produce or it will give you referenced genes or reference genomes from where this sequence is available. So that is your specificity. If your primer is very much specific for a specific genome or specific individual, so when we do the blast, it will not give you many genomes. It will give you a specific genome that this primer is specific for this specific genome or target genes. So you were getting my point, na? This blast for specificity and primer for design. Now, primer we are designing primer, and blast means it should be specificity. We will check the specificity, and then we will use this website. You all know this NCBI website where we have a tool called primer blast. Then it will be done later. So primer output interpretation. So whatever the primer will find out, finally whatever the output it will give, it will be reading result table, FNR sequences, TM, GC percentage, and product size. So we'll do these things later, and then checking for dimers or secondary structure. So we'll check the dimers or secondary structure is present or not. This is automated. So we'll not do it by using this hand or using by our suppose clicking something or not. It is already automated by the NCBI primer blast. It will check for dimers or secondary structures, and it will remove those primers which contains dimers and secondary structures, because dimers and secondary structures will create problem during our PCR amplification. So it will remove those things, dimers and secondary structures. And then you have this FR sequences, this one TM and GC percentage and primer pair. Finally, you will select the primer pair. Then in silico validation, finally we will verify. So verification is another important thing, because why we are producing these primers? We are producing these primers for suppose to amplify a specific gene sequence using the PCR. Na? So suppose if you have to do this whole activity, you will need lots of time or investment or money and all those things. Na? So the funding problem and all those things. So before spending any amount or any money into your research project, or before doing any kind of this, you can say wet lab activity, any kind of wet lab activity, or suppose extracting the DNA sequence and everything, na, whatever we do in our laboratory research to do PCR and finally amplify the gene, and then we'll go into another genomic research or genetic research, so or mutational research and all those things. So without doing anything, you can just do what you can do. You can use a laptop, you can use the NCBI database, and you can retrieve a specific DNA sequence, and then you can use NCBI this primer blast to design your primer, and then you can verify that primer also for specificity for the target genes. Whatever the genes you will do later, you will do your activity later in your wet lab, but before that, in silico validation means you can do in your computer with your this computer system, with your NCBI database. You can verify the specificity and target of the binding. Suppose you have developed a primer, and after developing the primer, when you are verifying it, you found that this primer is not specific or targeting my specific genes. It is not binding with my specific genes. So that time you can avoid that primer without going into lab, without doing any kind of this wet lab activity. Just using this primer blast, you can avoid such primers which is creating problems. Primers in the specificity and targeting, and then you can visualize primer binding sites. So with this you can visualize also. You can exactly see where this primer is going to bind in your target genes, because when you put everything in your PCR mixture, so you cannot track, or you cannot find, or whatever, you will only find the output. And if that output is not good, your whole PCR or your whole time and money, whatever you have invested, that will go on in vain. Na? So it will be not in use. So the point here we are discussing is that with the NCBI primer blast, you can verify your primer specificity and targeting, and also you can visualize primer binding site and compare across related genomes. And finally, what is the most important or what is the most useful things we can do with the bioinformatics is that whatever the primer you have developed, you can search it, or you can compare it across the genome, because in NCBI you have thousands of genomes. So with that you can compare. So see, common mistakes and troubleshooting. These things you just remember these things, because when we do hands-on activity, we have to avoid this type of mistakes like too high, too low GC content. Your GC content must be optimum, means 40 to 60%. It should not be very low, that is less than 40%, or it should be very high, more than 60%. Then we have to avoid primer, dimers and hairpins. Our primer sequence must not contain any kind of dimers or hairpins. Now we have to avoid these dimers or hairpins. Then incorrect target organisms. So sometimes what is the problem, that we will target organisms which is not our objective, or suppose you have produced or designed a primer. Now when you are targeting this primer into the genome, it is targeting a different organisms altogether. So your primer is basically not for your target organisms or the target genome of that organisms. So in that way also it is useless. And overly long amplicon size. Your amplicon, if your amplicon is very long, that is another problem. Now your amplicon must be shorter, means 15 to 24 base pairs, and 4 specificity settings. So your specificity must not be very poor. Now it should be very much specific. Then you have what is the application of primer designing? So it is disease diagnostics, gene expression analysis, mutation or SNP detection, and plant breeding and marker assisted selections. So these are our use or application of primer designing. So in these fields you can use the primer designing: disease diagnostics, gene expression analysis, mutation or SNP detection, plant breeding and marker assisted selection. So beyond primer blast, see, primer blast or NCBI primer blast is one of the tool with which we can do or we can produce primers. But there are many other different tools which we can use to produce primers, and those tools may give you more appropriate or more specific. But NCBI primer 3, after integrating this primer 3 with blast in NCBI database, now this NCBI primer blast is, I think, it is giving you the best result or best way of designing a primer. And there are many other softwares or many other tools available which we can use to design a primer. So summary for today's this presentation, what we have already discussed about theory, is that bioinformatics bridges biology and computation. So we will all know that what is the importance or what is the future of bioinformatics. Even today, at present, this whole life science depends on whole life science, which I am not saying whole life science means, because there are many taxonomic study and other things, but in a way our genomics and molecular biology studies basically depends on this bioinformatics tools. So if you are not using properly these bioinformatics tools and all those things, your result or your research will get impacted, or you will not find or will not give you the output what is expected from the research. Then primer design is the key bioinformatics application. So since we all know that all of the genetic study, genetic research or molecular biology research depends on a specific target gene. If you all agree with me that whenever we are talking about any research activity on genetics or molecular biology or any of these related fields, so the first and foremost thing come to our discussion is what is the target genes. Target genes or traits. So traits are related to the target. We all know that in genetics, all these traits are connected to the specific genes. So every time it comes to our mind that what is the genes. So it's in any time we are doing research or any traits we are studying, so our primary thing is our target gene. after amplifying this target gene, only we can do any kind of molecular or genetic study or mutational study. Nah? So that's why it is written here that primer design is the key bioinformatics application, because it will help you to multiply your target gene. And if you have multiplied your or amplified your target gene, means from 1 to 1000 copies or 2000 copies of your target genes, then you can easily do your research on molecule. Then many things can be done. There are many fields of research which only depends on this PCR amplification. Even your forensics. In forensics or in this criminology and criminal studies also nowadays. So everywhere, the first and foremost thing is a primer, because with this primer only you can amplify your target genes. And after amplification only you will get a proper amount or quantity of your target genes, which you can use to do many kind of research activities. Then you have hands-on learning improves concentration. So when we do hands-on learning, it will give you a more proper way or a more proper basis of today's this discussion. So I will not go much into the detail about this theoretical part. So if you have any question, you can discuss in the chat box, or any theoretical question if you have. And one thing you all have to do is, I have already shared one step-by-step guide, or you can say step-by-step guide, or method means there are some process with which you can design a primer using a primer blast of NCBI, primer blast of NCBI. So if you have to go through these steps which I have provided, this shared in your group, training group, already shared in your training group. So one thing you all can do, you can just see the steps one by one, whatever the activity, whatever the steps there in the hands-on part. And another thing you can do some basic research on your what is this NCBI database. So with that you have to open the NCBI database. So the first thing I will request you to open the NCBI database in your whatever the system you are using. If you are using your mobile, it will be difficult, but those who are using laptop or computer, you can open the NCBI database. So two tab you have to open. One tab is you open this PDF file, step-by-step guide, which is shared by the organizer to you in your WhatsApp group. And another thing you can do, you can open a NCBI database. So now I hope some of you have opened this NCBI database. I will share the screen, then only you will find. So Zakaria, please check.
Sir.
Shared or not?
Yes, sir. It's visible. Yes, sir. It's visible.
It's coming.
Yes, sir.
Okay. So I have to open my PDF, which already shared with you. Step-by-step guide, or whatever I say. So see, the first step which we have to do, we have to retrieve a target sequence. We have to retrieve a target sequence. So for this purpose, I am using, suppose You can use any target gene, but for today's purpose, I have selected this one, MCC gene. It is there in your PDF file, which is already shared with you all. So in this, you select gene. You follow what I am doing in the NCBI database and click search. So see, these are so I have simply searched MCC. I have selected the criteria as gene, field as gene, and then I have put MCC in the search box and then click the enter button, and it gives me some results. See, search results. So there are two MCC genes already there in your NCBI database. So it is showing. So I will use only the first one. See, the first one you can open in your laptop also. So here it is written as Homo sapiens. This is human MCC gene. There are other MCC genes are there. So I will use the first one, which is your WNT signaling pathway Homo sapiens human MCC genes. Click on this. Now see, after clicking on this gene, so here is the all the information about this MCC is there, official symbol, official full name, primary source, related source. So everything you can find here. Now, and then see here, these are the genes, exon, intron regions of the gene. So what I will do, see in the right section, right hand side, there is a table of content. There is a table of content. So in this table, okay, if I can read the pointer. These settings are not working. Okay, no problem. So in the right hand side, this table of content, so in the table of content you see in the right hand side, here it is reference sequence gene. So I will click on the reference sequence gene because we need the reference sequence gene of this MCC. We needed the gene sequence actually. Whatever you can say, it is reference sequence gene or the gene sequence. So if you click on this gene sequence, reference sequence genes of this MCC, so it will open like this. And see, this is the MCC gene. See here, Homo sapiens MCC regulator of WNT signaling pathway reference sequence gene of chromosome 5. So in this above, if you see here is you have a FASTA written just below, just the first line above. You can see the FASTA. Everyone, you can follow me. Are you following me? This FASTA written here in the top left side. After opening, just below the gene, see here the FASTA is written. So you will click on the FASTA. Now this is the gene sequence. See, this is the gene sequence, whole gene. It's a very long gene. So this is your MCC gene. Everyone getting my point, na? This is your target. This is the whole gene, or you can say this is your whole target gene of MCC, and this target gene is present in the whole genome. So when we are producing a primer, when we are creating a primer, you need a primer which is from whole this part of this primer is basically nothing but part of this whole gene, and which can target or bind with this gene during amplification of genome of human genome amplification. Suppose you have the whole human genome with you, so your primer will directly go and it will bind with this MCC gene present in the human genome. Are you getting my point? So primer is a part of the gene, and this gene is part of the whole genome. So if you are producing a proper primer, so during PCR amplification, this primer will go and it will directly bind with the gene which is our target gene. In our case it is MCC present in the human genome. So we are going like this, and this is in the chromosome 5. See, it is in the chromosome 5 this gene is present. So up to now, any question from anyone? Still we are retrieving the sequence. We have not retrieved yet. We are still retrieving the sequence. So our first part of primer designing, we have to retrieve the sequence. We have to find the sequence, our target sequence. So from this, you click on the gene bank written here. See, gene bank. You can follow the cursor wherever I am clicking. See, this is the information you get. You can read this information. This information are about the specific gene only, but our purpose is to retrieve the sequence. So for retrieving, you can see, analyze this sequence. Here is a tab in the right side that is analyze this sequence. In this analyze sequence, this highlight sequence feature. If you all can see, there is a tab highlight sequence feature. You click on the highlight sequence feature. So in the highlight sequence feature, this is your whole DNA, the whole target genome. So just in the lower side, if you see, just lower side, see, from here you can select your exon. We all know that there is a two region, exon and intron. So exon is basically the expressing part, or that is the useful part of the gene, and intron we know that it is in between part of this exon. So our target primer should target the exon part, because exon is the part from where this mRNA and the other activities or actual expression of this specific gene is going on. I am not going into detail of intron exon, because we all know this, what is intron, what is exon. But what we are saying here, that this gene contains both intron and exon. The long gene sequence of MCC contains both exon and intron. So we have to create a primer which can target the exon part. Suppose if you click give me 2 minutes, I am receiving one call. Go through this part. Up to now, what we have done is that we have a specific whole gene. Okay, I'll switch on my video. So up to now, we have a whole MCC gene sequence, which is a very big gene sequence, and means it contains lots of nucleotides present. And out of these nucleotides, we have to select the exon part, because we know that any gene contains both exon and intron. We have to select the intron. We have to select the exon part, and our primer, the target primer, should target the exon only. Now, any question you can ask. If there is any confusion. So what I am saying, that any gene contain both intron and exon. So like in our MCC gene also, both intron and exon. Out of this, so what we have did is simply NCBI database. So in the NCBI database, we have searched for MCC gene. MCC gene means you can use any other gene also, whatever the target gene. And the organisms which we have selected after MCC gene, there will be many MCC gene will come, and we have simply selected this one, human MCC gene. And after that, we have clicked on the FASTA format, means the sequence will come out. And from this sequence, we have done this analysis of the sequence. And analysis step, if you are clicking the analysis step of this sequence, you will find in the below region, in the below, see, there is a tab will open. It's opening, CDS, exon, gene, all these things are there. So we have selected the exon. Why we have selected exon? Because we all know that in our target gene there must be some intronic region, there must be exonic region, and our primer must lie nearby to the exonic region, because we have to target the exon region, because exon region is the region which will finally get transcribed and finally do the expression part of the specific gene. So if you see this page, in this page, this is the gene and all those informations are there, and just below this, if you select this exon region, the skin is visible, na? Current skin.
Yes, sir. It's visible.
So it's so if you see here, there is a feature option in the below tab. In the below side, there is a feature option. So in the below this feature option, you select the exon. I'm clear, na? So this exon, if when you will select this exon, you will see here 1 of 19. 1 of 19 means in this gene there is a 19 number of exonic regions are there. Simple. So when we have selected this analysis feature, which is your exon, so it is giving us 1 of 19 means this is 1 of the 19 exon. So suppose our target is the second exon, exon 2. Now, suppose we are targeting exon 2 of the MCC gene. You are getting my point, na? There are 19 number of exons which we are getting from this MCC gene. Out of this 19 number of exon, we are targeting, suppose we are targeting this exon 2. Targeting means we are trying to create a primer which can bind with this exon 2. Simple. We are trying to produce or trying to create or design a primer which can bind with this exon 2. So we are selecting exon 2, and in the right side you will see this FASTA option. So in the left side we have already selected exon in the feature region, and 1 of 19 out of 19 we have selected second exon 2 of the MCC gene, if you can remember, exon 2 of the MCC gene. And in the right side you will see this FASTA, display FASTA. So display, if you click on the display FASTA here in this side, right side, so it will give you the FASTA region. See, this is your FASTA sequence of exon 2 of the MCC gene. That is our complete explanation. So this is the FASTA format. This is the exon 2 of the MCC gene in the FASTA format. So adenine, thymine, so AIT, this is the sequence. This is the actual sequence of the MCC gene of exon 2. You remember that we are not targeting the whole MCC gene. In this whole MCC gene, there are 19 exonic regions. We have already avoided or already discarded this intronic region. We have selected only the exonic region, and out of this 19 exonic region, we have selected only this exon 2. Now this is the retrievation part. So we are still retrieving the sequence. We have not started designing the primer. We are still selecting our target region. So after getting the target region only, you will do the selection of primer. So this is your MCC gene, and in this side, if you see change region shown. So in this says selected region 108241. So this is the number of nucleotide from 1,08,241 nucleotide to 1,08,485 nucleotides. So this is the length of this exon 2 of the MCC gene. So now what you have to do, you have to open a Word file. Now is the designing part. You can say now is the designing part. So what you will do, you will copy this part. This exon 2, just copy it. Exon 2, just copy it. And open a Word file. Open a Word file. So I am opening a Word file. Wait. 1 minute, I have to share the screen also. So this is my Word file, and this is your Axon 2. So you have to just copy this Axon sequence, FASTA format, in FASTA format, open your Word file and paste it here. You can check this normal formatting, that if there is any gap or gap is present, so there must not be any gap because this is a continuous sequence. So this is your Axon 2 region. Now we have to design a primer which falls in this region or nearby this Axon 2 region of the MCC gene, so that our primer can bind during PCR. Our primer can bind to this Axon 2 region. So for this, another issue is there. So that issue is that whenever you have create primer for your target gene sequence, suppose here we have Axon 2, this is your Axon 2, suppose you can write it here. So this is your Axon 2 of your MCC gene. This is your Axon 2 FASTA format. So whenever we develop primers, so one problem generally we get is that if you select only this region, Axon region, only this Axon region, so the initial few part or few nucleotide of this Axon region will be discarded during our primer formation or primer development. Discarded means it will be not considered for primer designing. So similarly, in the backward direction also, there will be some nucleotide sequence which will be not in use during primer synthesis. So one thing we can do, we can add some sequences in the first part. Suppose this part, if you add some sequences here in the first part and some sequences in the last part, then only you will get a primer which falls exactly in the intronic region. Now you are getting my point now. This is your completely a proper axonic region. So what I am suggesting is that if you add some intronic region in initially and in the last part, so your sequence will be contain both intron and exon. Intron and exon means some introns and the main Axon 2, so that your primer can start in the intronic region and it ends in the your axonic region, so that none of your this nucleotide sequence from the axon is discarded or avoided during the primer designing. So what we can do, you can add some sequences in the initially and some sequence in the C. If you come to this right side, this is your change region shown. So in this, if you see, I have zoomed it. If you can see, so in this region, so you can add around suppose 200 more number of nucleotide. So if I say it is 0, so it will add 200 more. If I write it 0, so it will add 200 more nucleotide initially, and in the last part you can add 100 more. So suppose it was 4, I have now given it 5, means 100 more nucleotide will be added at the end, and 200 more nucleotide will be added in the beginning of your exon 2. So any question or any confusion here? So what we are doing, our exon 2 is okay. We have exon 2 sequence or in the FASTA format. But what we are doing, we are adding some intronic region in the beginning of this exon 2 and some intronic region in the end part of this exonic region, so that our primer completely falls in the exon only. So if you click update here, so it will change your FASTA format of this MCC gene is now bigger in more in number, more in number. So around 200 in the beginning and 100 at the end. We have added to around 300 nucleotide to the MCC gene. So just copy it and paste in the Word file. Now see the difference. I'm just normal formatting this. I'm using the technique which you all can repeat. There are many other ways to design this, but this way I think this can be done, or this can be practiced by you all in your own laptop. So if you now we have to find out this exonic region in this long exon intron region. So just copy this region. Just copy some nucleotide of this region. Suppose initial and Control F. If I paste, see, this is the starting point. This is your starting point. Suppose this is the starting point, and from the end also similarly. This is the ending point. So this is your original exon. This is the original, same as the above. This is same as the above. What we have done, we have added around 200. This is your intron. Suppose this yellow part is your intronic region. This yellow part is your we are still working in the Word file. This yellow part is your intron. See, now you can delete this part. No issue. So now we have exon 2, which is yellow, and at the beginning of this exon 2 FASTA sequence, we have added around 200 intron nucleotide and around 100 nucleotide at the end. So now you have a big long FASTA sequence of exon 2, which also contains some intronic region at the beginning and some intronic region at the end. Why we have added these introns? Because when we start this designing or start this designing this primer, during that time your whole exon, this is your whole exon, this exon part will be considered. Otherwise, for consideration, it will suppose avoid some part. Suppose if we have not added this pinkish color intronic part, so during your primer formation, this green color exon, suppose this green color, or there may be some other regions, from the beginning will be not considered for primer designing. Because whenever we put any this sequence for primer designing in the primer blast, so primer blast will few nucleotides from the beginning. So for this purpose only, we have added this one. We have added this intronic region at the beginning and some intronic region at the end, so that none of the exonic part will be avoided during the primer formation. So now we are ready with our sequence. Now see. Now as per your this one, as per your sheet which I have provided, see in the beginning you have a link for primer blast, or if in the Google also, you just write down primer NCVI primer blast and click on this first one. So this is the NCVI primer blast. This is the website. Website link is given in your PDF file. So you can open this link, which is your NCVI primer blast. So this is the site for creating your primer, for producing your primer. So now what you can do, you have your sequence retrieved in your Word file. So from this Word file, you copy this whole intron exon region and just paste here, enter session GI FASTA sequence. Just paste here. You can correct this some problem. So this is your MCC gene, exon 2, along with some intron. So this I have copied here. And see, here is the setup. Now you set up some parameters. So most of the parameters are okay, but two things you have to understand. One is your forward primer and another is your reverse primer. So in the forward primer and reverse primer, you can keep it blank also, or you can give your forward primer must contain which of this region. So this is your, suppose this is your region. So this is your sequence. So in this sequence, your forward primer contain how much part. Suppose this is the part which you want to put in your forward primer. Now this is the part which you want to put your forward primer. So if you see, this is from 1 minute. You just select beginning part. So this is 1 to 95 up 95. See. Now I have come to my so it is kind of, suppose 179 or 180 means whenever you are selecting your forward primer, you have to give the range from which nucleotide to which nucleotide you need your forward primer, and from which nucleotide to which nucleotide you need your reverse primer. So whenever you are selecting this forward primer and reverse primer, always include both intronic region which you have added to avoid any non-consideration of your exonic region. So whenever you select some part from whole part of the intron plus some part from the exon. Suppose this much I have selected, and then here you just need to give the range. Suppose from the first nucleotide to suppose 79 nucleotide. And for reverse primer, again, for reverse primer, suppose from here to here. So So first you check up to now is how much. You have to put some only the nucleotide number, nothing else. So whatever the nucleotide, suppose up to there. So this is a 430, suppose. So I am writing from 430 to the last nucleotide. Total number of nucleotide here is 552. So you put 552. Any question during this you can ask if you are repeating the same process. So I have just provided the forward primer range from 1 to 179 and reverse primer range 413 to 552. That means this green part, suppose from here. Suppose this two green part. Now this green part contains both intron and the sum of the exonic region. Similarly, in the reverse primer also, green part contains both intron and sum of the exonic region. Then something much you have to change. So see a PCR product size, it should be 30, maximum it is 10. Here you can give the number of primer you want. See here it is written 10. So if you want more number of primers, you can give 20, 15 or minimum suppose 2, 3. So suppose keep it like this, that we need a 10 number of primers out of this. Nothing you have to change much. Only thing you have to change is this one, RNA sequence. So in this you have to change the database. Database means it should be NT. NT means it will contains your reference sequence or any other form. So whatever the form of the sequence and rest of the things, it is Homo sapiens and other things, you keep it as such. So this is the NCPI primer blast. Here you have to put your, first you have to put your accession or the genome sequence, target genome sequence. In our case, we have inserted the MCC gene sequence along with some intron and exon regions. Then you have to write forward primer and reverse primer range. So I have given forward primer 1 to 179, reverse primer 413 to 552. Now, and 552 means total we have 1 to 552 means total we have 552 nucleotide here. So out of this 552 nucleotide, our initial range will be 1 to 179. Why 1 to 179? Because 1 to 179 nucleotide contains both introns which we have added plus the exon part. And similarly, this last part or ending part must also contain exon and the intron part. Now, just click get primer. See here, get primer. One minute. They should be the same. I think some of this sequence got deleted. While I was adjusting, I think some of the sequence got deleted. Again I am inserting the same thing. And total there are 545 total nucleotide. 548. So maximum it should be 548. 545. So there are total 545 nucleotide actually. So it was my mistake. Rest is same. Nothing have to change. Then select get primer. Now it will give you the result. It will take some time, very less time, but it will give you the result. See, this is the part. So in this result, see the good thing of this primer blast, SCBA primer blast, it will give you the exact MCC gene of your particular organism. Suppose in our case it is human Homo sapiens, na MCC regulator, and also if there are some Homo sapiens other coding genes, na clones present in the NCBI database, it will also give that also. It will also give that also. So we will select only this MCC gene. See, Homo sapiens, and submit. It will take some time, means few seconds only, and it will give you the primers. See here, making primers specific to your PCR templates. Our PCR templates, we have already given this template. That this is our MCC gene. We have added some introns in the beginning and at the end, and now we have given it forward primer and reverse primer. Forward primer includes we have put the instruction that our forward primer must fall between our first nucleotide to 179 up to 179 nucleotide, means our forward primer must fall in the intron and exon region, and reverse primer must enter into the must fall in the intron and exon region added in the end. I am getting back to this part, intron and exon part. Now see, these are your primers. See, the first one is your graphical view of the primer. This is your primer. Primer 1, then primer 2, primer 3, primer. This is the graphical view, and this is your forward primer, this is your reverse primer. In between, this is the gene of our target gene. So similarly, here is the detail. So this is your primer 1. See, forward primer. I will copy this. This is your forward primer. Anyone having any confusion, you can ask. This is your forward primer, just provided by our NCBI database, and this is your reverse primer. Now just check where it falls in your original input. This is our original input. So in the original input, I will just check our forward primer. See, just copy and Control F. See, this is the region. If you are getting very interesting, this falls. Yeah, this is the region of your forward primer, green part, and the reverse primer. Reverse primer is, see, this reverse for this searching. If you search it directly, it will not find. It will not give you any result. So it will give you result only after using another software or website. This one, reverse complement. This reverse, see, I have already shared this website. So you will have to open another tab where you have to search reverse complement. And the first reverse complement. So here you have to copy your reverse primer in this, submit, and it will give you this. Now this copy and again Control F. Now see, I have not copied it properly. It basically find it from GCC. Actually, if you reduce somewhat, see. This part, GCCT. This is your reverse complement. This is your reverse complement. Not up to that. So, in this here, if you observe your forward primer, so this is your forward primer. Now see the characteristics of your forward primer. See, here it is length is 24. Now initially it will start with 15 PSPI, it will end with the 38 after. TM is 57. GC content is 37 degree. Self compatibility, so this one. And again reverse primer is this one. 20 base pair long, 504 to 485 and 59 and PM. So these are some of the characteristics. And then again, so you can see for this primer one, the primer one which we have selected here. So for this primer one, these are your detailed report of the primer one. So from this detailed report, we just nothing much to use for our this preparation of primer, designing of primer. You have to just select the forward and reverse primer. So this is our forward and reverse primer, which is total 490 space here we have already used. So this is your forward and reverse primer. Now using this reverse and forward primer, you can now design. This is already designed. So using this forward and reverse primer, now you can use you can synthesize multiple product synthesize of these primers from the there are many institutes or there are many companies are also there who are working. So, or you can synthesize in your net to get the capacity in your lab. And finally, you will develop the primer which you will use for the PCR application. And another important aspect of doing this, there are another website that is your primer stats. In this primer stats also, simply open this primer stat website. We are getting late, so I will conclude. So this is where you have reverse. Suppose you have forward FWD forward primer and you have reverse. So just copy paste from this data types. Nothing much to do. So this is your forward primer and this is your reverse primer and submit. Then you will get detailed report, and this detailed report is directly useful for your PCR setup. Those for setting up your PCR for using this primer. So with this whole general result from this primer sets, the whole general properties of this forward primer and general properties of this reverse primer. See, here you can see this suitability test, single base pair run, pass. See, here it is a warning that it contains less than 40 DC content. So that means we have to again recheck or retry another primer which may contain more than 40. Still, in our case, it is only DC content is nearby 40%, so still it's okay. But still, it's showing there is a quantity that it is DC content is less than 40. So these things we have to understand from this primer stat software. So primer stat software or website will give you a detailed report on the primer design. Whatever the primer we have designed, it will give a detailed report. And based on this detailed report, we can check. You can check the feasibility or suitability of the primer by checking if it is pass. There is a warning. So all these things we have to see. Here warning of TM is greater than 58. So again, this is again the TM is higher side. So similarly, for your reverse primer also, for your reverse speed, in the reverse primer, the GC content is 55. If it is visible, so GC content is 55. So again, it is good because we need GC content 40 to 60%. So in this case, in the reverse primer case, it is 55. So that's why percent GC is written as pass. Since our GC content is 55, which is in the acceptable range, so it is showing percent GC is pass. And similarly, in this case, GC content is 37 degree, so it is showing percent GC is 40, which is 1. So there are other tools. The main initial tool, the main tool is your primer brush, and there are other this reverse complement or this TCF sets you can use to analyze your primer. Now, whatever the final primer you have developed, already your primer brush will analyze your primer very much in detail. Still, you can use many other tools to analyze. And see here, you have lots of primer that we have designed now. So, primer 1, then you have see primer 2. So, likewise, you get 10 primers because we have put that we need 10 primers. So, there are 9 to 10 primer used. In this case, it is 9. So 9 to 10 primer, possible primers already. Your TCF brush already provided us with those primers. So with this, I will ask you to put any question. Question means if you are using this tools, then you will find you may find some problem or you have some issues doing it, following whatever I have done here. And in any question related to the theory part also you can ask. And practicability, which I already discussed, that there are many practical aspects. And most of the people who are doing wet lab experiment on your suppose genetics or mutational studies or suppose forensic department, so they already know what is the use of this primer. Now the part of bioinformatics is that there are many bioinformaticians, or today in our today's life science students are more and more getting into this bioinformatics, because now we need to produce or design primers which we can use to amplify our target genes. Because target genes earlier it was like traits, and we link those traits with the gene. Earlier in the old conventional method of plant breeding or genetic studies, so we only observe the traits, and we know that there are some genes which is responsible for these traits. So those genes are not visible to us. But nowadays, since we are doing more research on genetic studies and more molecular things are there, so we have to design primers to amplify our target gene, and after amplification only we can do the other experiment which is related to the target gene sequence. So any question from the participants or any observation you can say. And also I would like to again apologize to the organizer and also to the participants, because normally it is very difficult to follow if resource person is not continuously giving you this or there are many interruptions during the resource person talk. So today actually it was some old day in our college, so there are many guests and all those things. So because of that only, otherwise any query now or in future also, you can discuss with me in my Gmail already. I shared in my PPT. We can discuss.
Thank you so much, sir.
Zakaria.
Yes, sir.
Zakaria, you can continue. You can
Yes,
continue.
thank you so much, sir. The meeting is open for queries. If anyone have any queries, they can ask their question in the chat box also. So I think there are no queries.
Okay.
Thank you so much, sir, for your valuable time and the insightful session. Now we are going for the last of the session. And I request Ms. Malika Borua to end the meeting with a formal vote of thanks. Thank you to Ms. Malika Borua.
Thank you, Zakaria. Good afternoon, everyone present here. On behalf of Bahana College, I extend my heartfelt gratitude to all who have contributed to making today's program a meaningful and successful one. I would like to extend my deep gratitude to our distinguished resource person for taking out valuable time from your busy schedule and sharing your insightful knowledge with us. Your words have truly enriched our understanding. My sincere thanks to our coordinator for her cooperation and supervisor in smooth conduct of this program. I also acknowledge the valuable assistance of our faculty members who have worked behind the scenes to ensure that all arrangements were made efficiently. A special thanks to all the participants whose active involvement made the session interactive and meaningful. Lastly, I thank everyone present here for being a part of this program. Thank you. Have a wonderful day.
Thank you. Thank you.
Thank you. Thank you, sir.
Thank you very much. Thank you.
Thank you, sir.
Thank you.
Yes, sir.
Thank you, sir.
Please, good afternoon, sir. Please share your affiliation and, if possible, in WhatsApp number. I am Dr. Ahmed from Telangana University Head Department.
Oh, sure, sir. Sure, sir. Good afternoon, sir.
Can you put it in the message box?
Yeah. Yes. I'm writing it down. You need the affiliation and this email, na? Or contact number.
Sir, full affiliation with the WhatsApp number.
One minute. Yeah. I have shared in the chat box.
Sir, WhatsApp contact if possible.
WhatsApp contact. Yeah. Yeah, I've shared the WhatsApp contact. Also, everyone can contact me on the WhatsApp because I am available all the time, and we can discuss some research work also in future. Sir, you got my number, na? Abdul sir.
Yes, sir.
Yes, I have shared in the chat box. You got it, na?
Yes, sir, I got it.
Yes, sir. Thank you, sir. Okay, Zakaria.
Sir, okay, sir.